Okay, thank you very much. Yes, I was asked when I was asked to provide the title to the talk. I came up with this, and because we do some work with genomics, but it's not genetic manipulation. We're just measuring what's in there already. We are not changing it in that sense. There's enough to work to go on with. There's a number of challenges on the horizon or imminent. Really, we have to, because of a growing population, we have to increase productivity, adapt to changing climatic conditions, and we are also there was talk about Drexler, which is something that has gradually creeping up from more southerly latitudes, so more virulent pathogens, and an increase in plant pests and their ranges. So there's enough to work on for the foreseeable future. And the goal is to breed forage crop varieties that provide high quality nutrition for ruminant animals. So that's the for the time being anyway, yes. And that'll probably go on for a while, you know. And I'll start talking about perennial ryegrass, but I will also mention red and white clover, and I'll even show some of the same slides that Helen had. But never mind. And also go over some of the same ground that Alan has mentioned before. What are the breeding targets that underlie this overall goal? And there's biomass yield, the quality, and the heading date, and the DUS traits, the distinctiveness, uniformity and stability traits. Persistency is important, and of course disease resistance, resource use efficiency, and of course seed yield. If you can't achieve a significant seed yield, it's not worth breeding the variety. And then resilience to climate change, which is sort of intertwined with some of the quality traits that we are trying to breed. And how do we get there? There are 4 things that we can work on when we do breeding. We can change selection intensity, and you also need the trait to be heritable. If it's not heritable, forget it. You won't get any improvement. So the heritability is related to the accuracy with which you can increase the genetic gain. And then you also need to have a population in which there is phenotypic variation and heritable variation, because otherwise you will not, if there's very little variation, there's very little to select on. And fourthly, time is an important factor. If you can crank up the speed of the breeding cycle, you might be able to get to a new variety faster. And that's where the genomic selection and the markers possibly come in. And I'm showing an equation, and there's a famous quote from Stephen Hawking saying that every time you put in an equation, you halve your readership, so half of you go to sleep. But anyway, that just sums up those 4 things that make up the genetic gain: selection intensity, heritability, phenotypic variation, and then if you can reduce the length of time, you automatically increase genetic gain. And again, another slide that's been shown before, and the breeders that have used conventional techniques such as Alan and Sarah, and before him Pete Wilkins, and other breeders indeed have made a lot of progress over the last 50 years. So it's a difficult ask to improve that, and I'll come back to that. And now I'll go through a little bit of how new varieties are bred. And on the left, you can see the current selection or current breeding scheme, and you go through a cycle here every 4 years. That is because it actually only takes 1 year to complete. But then you have to, when you have your mother plants here that you take your seed from, you put in the plots, and you select or you assess all these different traits that we've been talking about, yield and quality and so on, and over a period of 3 years. And then you return to your mother plants and you base the data you collect here to inform which ones go through the next cycle. So that takes 4 years. And if we could crank up the speed with which you go through that breeding cycle, it would be helpful. And that's where a potential genomic selection scheme, where you use thousands of markers in one go to help you, and then you do your you put your plants in the field, and you do the polycross. That means the cross between loads of different plants in a chamber where you know the pollen donors, so you don't get contamination from outside. And then you go through that. You can do that in 2 years rather than 4. So that would help us to speed up the breeding cycle if we trusted the genomic selection. And that's the big question: can it compete with conventional phenotypic selection that has worked so well over the years? And this is how the principle of it. In fact, in principle, it's exactly the same as near-infrared spectroscopy that is used routinely in most breeding programs to measure quality traits. You need a training population, and you have where you measure phenotypic data, any one of the ones we have mentioned earlier, and then you also analyze the population for marker data, and you put those two things together, and you get a genomic breeding value, and you then feed that. The theory is you feed that into a breeding population where you only have information on the marker data, and then you use the genomic value to do your selection, and then you get your candidates for the next generation. So that's how it sort of works in principle. And how does it actually stack up at the moment? Now, that depends very much on your trait. I took two of the traits and got data from Alan and Sarah of phenotypic data for the dry matter yield in year 2. And then this cloud of dots represents over 5,000 progeny from simulated cross between. You have your latest generation consisting of 100 mother plants at a given point, and then you do all the pairwise combinations, and you get a little bit over 5,000. And those are the 5,000 points. And along here you have the mid-parent value, so the average of the two parents, the phenotypic value, the actual value that has been measured in the field. And then you compare that with the predicted value, and you would like to see a linear relationship, and you can see it's not quite like that yet. And then I went on to try and select the best 4 parents, which is what the breeders do when they make a new variety. The best 4 parents, only thinking of dry matter. And then when you do it on the phenotypic data, you are selecting on a very select group here. But when I did it without knowing the phenotypic data for that generation and just took the genomic value, the worst of the pairs that I got was all the way down here. And this is not, you're only avoiding the worst rubbish. You are not really selecting on the very, very best. So for dry matter yield is not yet as one would like it. The thing look a little bit better for the quality traits, and I've taken water-soluble carbohydrates, the high sugar trait, as an example. So the same here for the phenotypic trait. It's still a little bit better than the genomic value, but this is within sort of the same area, and so the genomic selection would work because you save 2 years. So it only, in theory, needs to be, the accuracy needs to be half as accurate as the phenotypic selection, or a little bit more. But so it looks a little bit more encouraging for quality traits. The biomass traits are a little bit more difficult to handle because they're more variable. I will just briefly go through a number of other projects, or a number of projects. All of them have actually been mentioned. There's the increase in seed yield, which is led by Sarah Palmer. Yeah, as I said, if you can't get enough seed, it's no point. And Magnet, looking at mineral nutrition and balance in ryegrass, leaf spot or drexler, and resistance to that. We're trying to breed into the high sugar grass varieties. And then the lipigrass project, which has just finished, and there is actually some marker work that has led to a potential variety with better fatty acid composition, but it remains to be seen whether the markers are having the required effect. And we are trying to get money for a continuation of this from funders. Then I'll go on to red clover, and also some of the same things that Helen was showing. We have some material that sort of carries on the theme from Aberclaret and Aberchianti, which are more persistent. And here's a new line here that David Lloyd, the Clover breeder, is working on. And as you can see here, after 5 years, it looks much better than your Merviot. So there is a visible gain in persistency in red clover that we are breeding into new varieties. So we're very hopeful with that one there. And then there's another project which is pending. We have applied for money, but we're still waiting to hear whether it's been awarded. So that's another use for red clover, where we're trying to extract protein from the forage and use it so that we can feed monogastrics like pigs and poultry, and so you get the protein out and you use it that'll be used in animal feeding trials. And then there are other products that you can use from these, from the screw press you get and the liquid you get out there. You get some peptides also that another company, Croda, is interested in using, and they're involved in it as well. And you get the fiber after that you can use to extract other high-value metabolites from the clover, and there's another Blue Sky Botanics involved in that. So we are very excited about that, but we're still waiting to hear whether it's been awarded or not. Then I'll talk briefly. How much time have I got? Okay. Another way, a little bit more sort of poor man's marker-assisted selection, but nevertheless definitely useful. Now, if you have the way the breeding is done is you usually have a mother plant with some seed. You're harvesting from it, and you know the mother, but the pollen has come from an unknown group of plants that have been pollinating this mother plant, and so therefore if you knew what the pollen donor was, you could use that information to increase increase the genetic variance that you have to work with. So if you knew the biomass yield of the pollen donor, that would be helpful. But in most, up to now, that genetic variance has sort of gone away and lost. So as I said, if you know the pollen donor, you can increase the use of genetic variance. And this is basically paternity analysis. You then use a few markers. You only need a few markers to reveal who the pollen donor is, and the mother you know. And so you get these patterns, and you need to put them together to get the paternity. So that is actually proving very helpful, both in the grass breeding program and also particularly in the red clover program, as I'll show now. We are trying to breed stem nematode resistance, higher stem nematode resistance into red clover. And in 4 generations of selection and crossing, which was also done in the Brimvar project, Matthew Lowe and Rhys Kelly have managed to increase resistance in families to over 50%. It's still a long, hard slog, but it does work. And the paternity analysis has helped us to get the families mixed most appropriately. So it has been helpful there. So finally, white clover, which is also part of our portfolio. And I'll just briefly mention the apple project, which has come up before, and work done by Collins and Lloyd and many others in that project to try to increase phosphorus use efficiency. Phosphorus is a finite resource, and some forecasts have said that, it's going to run out in 30 years or whenever, but or certainly high quality phosphorus fertilizer is going to run out, and the more efficient we can use the resource, the better in any case. So there are trials going on with that, and they look promising. And then I will also repeat a little bit about what Helen was saying about this promising hybrid everlasting, which originates in a cross between white clover and Trifolium ambiguum Caucasian clover, which is a close relative to white clover and is very drought tolerant and very pest resistant. And it has rhizomes rather than stolons, but it grows a bit too slow to be of any agricultural use. It is very difficult to get these crosses done successfully and actually get plants out of it. And the first successful cross was done way back in 1996 by Margaret Meredith, who's now retired some years ago, and her colleagues. So, and that's the slide from before. You cross them, and you need a method called a tissue culture method called embryo rescue, and then they finally managed to get a plant that was a genuine hybrid, and that has been back-crossed repeatedly to white clover, but still making sure you keep the rhizome trait, which is what we believe is responsible for all the good properties of this hybrid. And everlasting, the stolon networks are not as prominent as it is in white clover, and it spreads through these rhizomes, and it is very resistant to cold, pest, and drought and grazing. And there is a great deal of interest in it. So we're also quite excited about that. And here's a drought trial that was done, I believe, in the Sure Root project, where they were put in bins and not watered, and everlasting perform very, very well under these conditions. So it is a promising material. And then we have a few other projects ongoing. We have a big EU project that we are participants in. Myself, David Lloyd, and Rosemary Collins, to improve EU's and China's protein self-sufficiency. So that's basically involving 5 legume species, of which red clover is one, lucerne is another, faba bean is another, and pea is the 4th one, and then there's soybean. And we are one way or another involved in 4 of them, not in soybean. Smart expertise is a continuation of the disease resistance breeding that has been funded by the Welsh Government and EU. So, and that has just started, so we hopefully will be able to finish the breeding of nematode resistant and sclerotinia resistant material in that project. And then I'll just finish by mentioning a few people, or quite a lot of people who are part of the whole forage breeding setup. Sarah Palmer and Ella Lovett are the senior breeders of grass, and also Sarah is involved in tetraploid ryegrass together with Sheena Duller and John Harper, and Sheena is responsible for the amenity grass breeding program. David Lloyd is responsible for the clover breeding, and there are other key staff involved in various aspects of it. Is Rosemary Collins, Ratan Yadav, and Jim Vale is the big boss in the field experiments and planning everything and making sure it gets done, and loads of researchers and technicians making it possible that all this breeding can take place. And the Lovett is involved in the Leif project, Mark O'Farrell in national list trials that are also going on here. And in seed production, it's Ben Ling, Matthew Theobald, and Anthony Davies. And Danny Thorogood has just taken over from Jan to Thomas. And together with Lin Huang, they're splitting the responsibility between them. And with that, I'll say thank you for your attention. And also bring your attention to this day that on the 1st of May. Okay, thanks.
Does anyone have any questions they'd like to ask Brian? In John Innes Centre, they've developed they're called speed breeding, where they're doing like 8 generations a year by manipulating the lines. Is that a good thing, and is that something you'd be looking at here to do in IBERS?
Yes, we have briefly thought about it. Yeah, I mean, I'm sure it's a good thing for them, but I think it's less of an advantage here. I mean, we have links with New Zealand, and you could send seed there and get another season in, but it's logistically it's quite a difficult.
And it's a breeding methodology. They're looking at cereals.
Yeah,
where
it's
you're
very difficult.
Make crossing and want it made homozygous, so they need to go through several generations to achieve that. Because forages are outbreeders, you're not doing that, what we call purifying, if you like, steps. So we don't need to go through that number of generations to get to a variety, to get a homozygous or largely homozygous. So speed breeding is not the same advantage.
Yeah, we're actually selecting at each generation, whereas with cereal breeding will only select when it gets to the F5, F6, or 5 or 6 generations. They're not doing any selection at all. They're just getting pure lines together for selection in later generations.
Yeah, forage variety is a population of individuals that are genetically different, but a cereal variety is all individuals in that variety are genetically identical or very, very near.
Another question, Gary?
Hello. By using genomic selection, I guess you are fast-tracking the selection process. Are you also not fast-tracking sort of a genomic cul-de-sac, if you like? and you are getting away from some of the genetic diversity we might be looking for in a few years' time. We mentioned public goods and some of the traits we'll be looking for in the future. Do you think you'd be sort of breeding those out if you're breeding too quickly down one particular path?
You do have to be careful, that you renew your breeding population, because you do risk inbreeding depression, really, if you narrow the gene pool too much. So yeah, that is an issue with genomic selection, that you reach that point a little bit or a lot quicker than with other methods. But you will have to put in you'll have to inject new material into your breeding population then.
The other thing is the way we breed. When we're moving on into generations, we're trying to, in that section, we're trying to preserve as much genetic variation as we can, right? We're choosing families from the previous generation. The whole family goes forward, and that will include variation from the pollen donors in the previous generation as well. So you're getting a lot of, whereas when you're producing the varieties, they're an offshoot of the breeding program, if you like, and you're limiting yourself to 4 genotypes per variety. But you're preserving most of the genetic variation than the previous generation by using the families to.
Yeah.
Might not have explained that very well.
A new variety is more of a cul-de-sac, because that's 4 parents and you produce seed from those, and that's it. Whereas you are selecting for the next round from a lot larger sort of genetic pool, really. So.
A few years ago, when that's what his name now, was very nearly succeeded in getting nitrogen-fixing nodules to go on some barley. Are you, I don't know if it's here, but in the world, are we anywhere near getting nitrogen-fixing nodules grown on grass plants?
That old chestnut. No, I think not that I'm aware of. I mean, I think there have been people at John Innes who have been trying to put it on wheat, but I haven't heard of any success, and I'm sure we would have if there had been any. Bonkers, if you ask me, but yeah, that's just me. But it may be.
Bacterial endophytes,
yeah.
and some of those fixed nitrogen.
But they have been found in sugarcane, and I know an institute in Brazil who have been trying to prove that they actually fix some nitrogen to the sugarcane or contribute nitrogen to the sugarcane, and they have had difficulty with that. So I'm also a little bit. They do fix nitrogen, those bugs around the roots, but how much benefit it does to the plant is debatable, I think. So yeah, I'm skeptical about that. And I have worked with nitrogen fixation for 15 years before I turned to this. So I believe in nitrogen fixation in legumes. That is a beneficial trait of those that really should be used a bit more, but yeah.